the activity of, the action potential, excitatory presynaptic or inhibitory presynaptic excitation may go on. Okay, or EPSP potentials. I mean, sorry. So, in saying that, let's talk more about the action potential. All right. So let's say this neuron is being stimulated. Okay, it's being excited. Now, when we talk about the action potential, we kind of have to graph this out. Okay, we have to graph this out. Okay, so if we kind of draw a graph on the board here. All right, and we'll say this is, I'm going to abbreviate this, membrane potential. Again, think of that as the electrical charge. Okay, and this will be time. Okay, and I'm gonna put negative 90 millivolts, put negative 70 millivolts, put negative 55 millivolts, and then we'll put 0, and then I'll just put plus 30. Okay. And then down here, we'll do this in milliseconds. We'll go 1 millisecond, 2 millisecond, 3 millisecond. Remember, none of this stuff is drawn to scale. This is for the sake of discussion. Okay. Now, when we're talking about an action potential, okay, remember, neurons are irritable, excitable tissues. Okay, they're very, as I mentioned when I talked about glial cells, neurons are very sensitive cells. Okay, they're very sensitive to changes in the environment. Environments that you find them in. Like I said, whether it's a neuron, whether it's another neuron communicating, with another neuron, or it's a stimulus like a change in temperature, electrolyte balance, whatever it may be. Okay. Now, every time a neuron gets irritated, though, it's not going to fire an action potential. Okay. Neurons have to get irritated to a certain point. Okay. Typically, when we're talking about initiating an action potential, we start with something called a graded potential. Okay. Think of a graded potential as like a local irritation. Okay, a local irritation. All right. So let's say this is a mechanoreceptor. This is a receptor that's sensitive to touch. All right. And let's say I'm in a lab setting. I've taken this nerve out of some animal's skin. I put it in a solution, that's going to stimulate normal body fluid levels of that organism. Okay, and then let's say I've got my voltmeter hooked up to this. All right, and let's say I take a little glass probe and I go in and I press on this cell membrane right here. Okay, let's say I put just a little bit of pressure on there. All right, and what you're going to see is just a little bit of a spike. Okay, and then it'll probably go back down to normal. Okay, probably go back down to normal. Okay, that would be a graded potential, a little irritation. Okay, now what happened here when you see this up spike on here? Well, remember when we have that blow up of the neuron on there. Remember the sodium potassium pump. Okay, 3 sodium out, 2 potassium in. Okay, when you irritate a neuron, what's going to happen is that's going to trigger sodium gates to open a little bit, whether it's via a chemical message or a stimulus, but a sodium gate's going to pop open. Okay, and because there's more sodium in the extracellular fluid than the intracellular, sodium's going to diffuse inward. Okay, sodium will diffuse inward, and that's going to make the inside of the cell slightly more positive. Okay, so as a result, that's going to make potassium gates open a lot more, and a little more potassium is going to travel out. You'll have an efflux of potassium. Okay, but let's say this was a really weak graded potential. It didn't have that strong of a magnitude. Okay, I just irritated a little bit of that nerve. That was enough to open a few sodium gates, make a few potassium gates open. Okay, big deal. All right. So you'll notice that you saw a little up spike, and that was sodium in, and the down spike is potassium out. Okay. So now there's more sodium inside the cell than we need, and more potassium out of the cell. That's what the sodium potassium pump is there to do. Okay. It takes this excess sodium on the inside, gets it back out, and takes the excess potassium on the outside and gets it back in. Okay. Hence this little hence the, returning back to membrane potential. When we're at rest, the sodium-potassium pump, or right after an action potential, plays a big role in helping us recover. Okay? So that graded potential wasn't enough. All right? Or if a nerve synapse with this neuron and it released a neurotransmitter, okay, and let's say it wasn't enough to excite this neuron to the point where it generated an impulse. Okay? Now, so when it comes to graded potentials, how intense a graded potential is depends on the magnitude of the stimulus, okay? So now let's say I take this glass probe and I press even harder on this neuronal cell body. Okay, I press really hard on this, and and as a result, the stronger mechanical changes is going to stimulate more sodium gates to open. Okay, it's going to stimulate more sodium gates to pop open. Okay, so as a result, what you're going to see is now you'll see a bigger graded potential. Okay, now more and more sodium came in. Okay, and what happened was that the inside of the cell started to depolarize. Okay, because remember, when you're talking about polarity, you're talking about something having like an ion or a molecule having a charge. Okay. We call it, so basically remember the resting membrane potential is negative 70 millivolts. That's a charge, okay? So we say depolarization because, remember, the inside is very negative, okay? As you have more and more positively charged sodium ions coming in, okay, that's going to bring the membrane potential closer to zero. Okay, so as a result, that's depolarization. Okay, that's depolarization. All right. Now, like I said, I took that glass tube and I pressed on that even harder. Okay, I pressed on this neuronal cell body even harder, and you saw an up spike in the electrical charge in the membrane potential. Okay, so basically more and more sodium gates popped open, and enough sodium came in to the point where we hit this charge called threshold. Okay, in order for a neuron to generate an action potential, we have to depolarize it. We have to irritate it enough. We have to get it beyond this threshold. Okay, this threshold is important because we don't need a neuron to, waste energy sending out action potentials because every time it's slightly irritated. If that happened, these cells would become overworked and they would just shut down. Okay, so as a result, we need to in order to generate an action potential, we need to depolarize to threshold. Okay, and then once we hit threshold, okay, once enough sodium comes in and the inside gets more positive to this point, then that's when we start the action potential. Okay, and that's when the sodium gates of the axon hillock are really going to come open, and a lot of sodium is going to come in. Okay, so then what you're going to see, if you were to graph this out, you're going to see a big spike. Okay, you're going to see a big up spike, and then very quickly, almost as quickly as you see the up spike, you're going to see the down. Okay, then it's going to look kind of like this. All right. So what's going on here? Okay, so if I look at this little area of a neuron right there, okay, if I look at this little area right there, what's happening, okay, is remember, sodium gates are opening. Okay, so as sodium comes in, that's going to make the inside more positive. Okay, and so as a result, hence the big up spike. When you look at a scale like this, or a graph like this, that's what you're seeing, the sodium influx. Okay, you're seeing a big sodium influx. All right. Now, remember, these sodium gates are electrochemical gates. They're sensitive to changes in this electrical potential. So as a result, once the cell gets positive to a certain point, once it reaches a certain charge, that's going to make these gates coil up and close again. Okay, and then as a result, simultaneously, what's going to happen are potassium gates are going to open now. Okay, so then what you're going to see is potassium is going to go flying out of the cell, because as you remember when you learn about membrane potential, there's more potassium on the inside versus relative to the outside. Okay, think about it. The inside of the cell just became a lot more positive. Potassium is positively charged. Well, you not only by diffusion is potassium going to leave, but because of the same charges creates an opposing force. Okay, so potassium is really going to get forced out of there. Okay, and remember, the reason why sodium is not forced out is because the gates close once we get to a certain point. Okay, and remember, sodium is not going to pass through a potassium gate, so that sodium is just going to remain on the inside of the cell. Okay, and then so that's what this big downward spike is going to be. It is going to be a potassium efflux. Potassium is moving out. Okay, potassium is moving out. And a lot of times, and as the cell gets more and more negative due to this potassium efflux, okay, what happens though, not all the potassium gates are going to close all the way. Remember I said a lot of potassium gates are left slightly open. Okay, so that's going to cause a little bit excess potassium to leak out than what we want. Okay, and then what's going to happen is that cell is going to hyperpolarize. It's going to go beyond, it's going to become more negative than we want it. Okay, it's going to become more negative than we want it. But that's where the sodium-potassium pump comes in. Okay, this helps us reset or get back to a resting membrane potential so we can, so that cell can fire again. Okay, so the cell can fire again. So the sodium-potassium pump is going to pump out 3 sodium ions into potassium ions. Okay. So now one thing, so when you take a look at this, pay attention to the time, okay, and this should help you appreciate the sodium-potassium pump a lot more, because this happens fast. I mean, we're done with an action potential in about 3 milliseconds, give or take, okay? So that means, and that's the beauty of this, of having this resting membrane potential established, by having such a lopsided, amount of, sodium in the extracellular fluid versus the intracellular fluid. So when that sodium gate's open, okay, because there's such a steep concentration gradient, it's just going to fly into the cell. Okay, it's going to come in very rapidly. All right, and that's what, hence causing the very rapid depolarization, and that's going to make those sodium gates close just, almost as quick. It's going to seem almost as quickly as they open. Okay, and then same thing with potassium, because of this steep concentration gradient, across the cell membrane of potassium. Okay, again, they're going to open and they're going to close just as quickly. Okay, so there's still going to be a lot of sodium in the extracellular fluid because, like I said, since these gates don't stay open, for very long, okay, not all the sodium is going to come in. All right, that's why this is considered active transport. Okay, the sodium-potassium pump is active transport. It's actively pumping sodium out against the concentration gradient and actively pumping potassium back in against the concentration gradient, okay? And as a result, we have to burn ATP to do this. Again, that's why it's called the sodium-potassium ATPase. It's the enzyme that's used to cleave off the phosphate group of ATP so we can release energy, okay? Because remember, active transport takes work, and this is one of the biggest energy expending activities we do on a day-to-day basis, maintaining this membrane potential by using these active pumps, these sodium-potassium pumps. Okay, so basically what's going to happen then, we're going to depolarize. So then this active potential is, so when we drew this graph, this just happened in this one little area. Okay, because remember, we said that this cell is irritable, but we have to conduct that action potential. Okay, sure, we depolarize the axon hillock, but we have to do this all the way down the length of the axon. Okay, so what's going to happen then is as sodium comes in, as the internal environment of this area depolarizes and becomes more positive, that's going to be enough to irritate the adjacent side of the axon. Okay, and that's going to cause more sodium gates to open, but potassium gates is active potential. So when it comes to depolarization, it's not as linear as just we start an electrical signal here and it just travels, literally like it's going down a wire. Okay, what happens is we have these little local irritations, and these local irritations are just transferred down the axon. Because, like I said, this area gets irritated, that's going to cause the next adjacent segment to get irritated, and then the next adjacent segment. So basically, what's happening is while this area is depolarizing, this area is going to start very soon. Okay, this is what we call propagation. Okay, the active potential is propagated down the axon. Okay, think of propagated as it's transferred down.